All right. Good morning from Kansas City. It is 6:45, I think it is, Kansas City time, which makes it 4:45 LA time. So just a couple of things. If I sound out of it, that's partly why. The other thing is I found with these recordings that if I pause them, sometimes the audio loops back in a weird way. It only happens when I pause while I'm recording, not if you pause while you're listening to it. So I'm going to try not to pause no matter what. That means in about 20 minutes they're going to deliver my oatmeal and decaf, so you'll probably hear that. My husband is sleeping just adjacent to me, so if you hear snoring, that's him, but I'm just going to try to keep on trucking no matter what, so bear with me. Alright, so in this lecture we're going to talk about chemicals that cause cancer and then radiation as well, and talk about how they can cause cancer. To start out with, we did a little epidemiological data last time, and I want to show you some more specific epidemiological data associated with cancer-causing chemicals. 1761, Dr. Hill reported that snuff, which is this powdered form of tobacco that would be inhaled, was tightly associated with nasal cancers, and they showed a very clear correlation between the two all the way back in the 1760s. In 1770, a British physician noticed he had a large number of patients who had scrotal cancer. There's sort of these oozing sores that turned out to be a form of skin cancer specifically found on the scrotum. What he noticed when he talked to his patients is that all of them, or almost all of them, had been chimney sweeps when they were younger, years and years previously. And he postulated that some of the chemicals in the chimney and the soot were being absorbed by the oils of the scrotum and then eventually causing scrotal cancer. In fact, scrotal cancers were rendered nearly obsolete with protective clothing that chimney sweeps now wear and with good hygiene, washing after you are in the chimney. So that seemed to cut the rates dramatically. 1856, we start to see some real specific dose curves here. This is for 2-naphthalene, which can cause bladder cancer. This is a synthetic dye that was developed. This is right around the Industrial Revolution, where there were several new dyes being created. Before that, dyes were almost all natural products. And what they found is that people that worked in this facility, the employees, had an incredibly high cancer rate. In fact, in one of the factories that they looked at, every single employee of the 15 that worked there came down with bladder cancer. It's a really tight association there. Here you're looking at how long the people worked in that facility and their percent with bladder cancer. So for example, in red, if you worked in the facility for under 2 years, right, and then after your original dose was at time 0, here's your bladder cancer risk. And if you worked there for 3 to 4 years, here's your bladder risk. And if you were bladder cancer risk, and if you worked there for over 5 years, here's your bladder cancer risk. So really tight association showing the link between this particular dye and bladder cancer. Some of the early data showing that chemicals often found in the workplace can indeed cause cancer. So you know what the #1 most lethal commercial product is for causing cancer, and that of course is cigarettes, without a doubt. The second most lethal commercial product is asbestos. Asbestos was not commonly used until the early 1900s, when large amounts of this naturally occurring chemical were found in Canada, and then it was refined and was used for all sorts of things. It's a great insulation, for example, and so it was used for lots of different products. Unfortunately, what they found is that asbestos breaks down into sort of a fine dust. And if you look at the dust under the microscope, it really looks like a whole bunch of these little needle-like fibers, and that's really what asbestos is made of, are these tiny little needle-like fibers in this dust. So they started to see associations between asbestos contact and lung cancer, a particular type of lung cancer, in fact, called mesothelioma. Now, mesothelioma is a cancer of the mesothelial cells. These are the cells that form the inner lining of our body cavities, and they also line the inside of the lungs. I'm sorry, they don't line the inside of the lungs. It forms the lining of the inner body cavity as these mesothelial cells. So what seems to happen is when you inhale the asbestos, this dust, which has these tiny little needles in them, these little needles get lodged into the lungs after you inhale them, and then they cause inflammation because the long-term kind of irritation of the lung tissue starts to cause inflammation, and that inflammation eventually promotes the development of cancer in those mesothelial cells, that lining tissue. You can see the annual deaths from mesothelioma rising. This was after exposure. It takes about 10 years or so to develop mesothelioma from asbestos. So we can see a dose-response curve rising after that amount of time of contact with this particular carcinogen. The other question that lots of people have, and probably rightly so, is environmental pollution and its possible association with cancers. Now, pollution in the environment was linked to possibly causing cancer because of the seeming increase in cancer cases with the Industrial Revolution as the industry developed, more and more factories making various different synthetic compounds, spewing waste into the environment, and it seemed to correlate with an overall risk in cancer cases. And so like the two happened together. So the correlation was hypothesized that perhaps the pollution in the environment was responsible for these additional cases of cancers. So what they did here is they're looking at age-adjusted cancer rates. Remember that older people have a much higher cancer risk, so you have to look at age-adjusted data. But if you look at age-adjusted cancer rates, they are about the same as 100 years ago, before the Industrial Revolution, except for one type of cancer, and that's lung cancer. We know what the major correlation is there, that's cigarettes. So here, they're showing you the rise in pollution coming from pesticide production, right? And they're showing you the rise in pollution coming from plastic production, huge increases here that we see, kind of these contaminants spewing into the atmosphere. But if we look at two common forms of cancer, in this case we happen to be looking at women, but it's actually very similar if we look at men and if we look at other types of cancer, breast cancer rates and colon cancer rates really haven't changed dramatically over that time period. So we really don't have data to back up the idea that pollution in the environment is causing a significant number of cases of cancers. Cancer rate did go up, but cancer rate went up specifically in lung cancer, and we know that the correlation there is for cigarettes. Excuse me. Good morning. Maybe right here for a bit. Will it fit? If not, we can use the table.
And there's 18% gratuity.
Okay.
3,000 or 4,000 will call it.
Okay.
Is there anything else we can do for you guys?
I think that's great. Thank you.
Have a great day.
You too. All right, sorry, that was my oatmeal and coffee. All right, so where were we? So it looks like environmental pollution is not a significant risk factor for cancers, at least not has been identified so far. Another question that's been asked several times, a lot of the studies that have been done on individual chemicals and their ability to cause cancer has been done in animals, for obvious reasons, hopefully. Right? It was done in animals because you obviously cannot simply infect people with cancer or cause cancer in humans. So most of this work has been done in animals, or it's epidemiology work that's been done. So if you're going to do this work in animals, you have a limited budget and limited time frame, so you're likely to try and get this study going as quickly as possible. So what I'm telling you is that in order to get reliable data quickly in animal studies, animals are usually given what we call the maximum tolerated dose of whatever the chemical is that they're testing. Now that maximum tolerated dose is the maximum that they can give that won't immediately cause death in the animal or won't immediately drastically affect the animal. So what I'm telling you is they give them huge amounts of these chemicals in order to look for a cancer risk. Generally speaking, that is much higher levels than we would ever have contact with as humans in the environment. And that's just so that they can get their work done as quickly as possible. They want to make sure they've exposed enough animals to the chemical in order to get good data. So, if you look at the graph on the left here, if they're showing you data in green that comes from animal studies where they give these huge levels, the maximum tolerated dose of the carcinogen, and sure enough, they're seeing a correlation with tumor yield on the y-axis with the dose of the carcinogen. And what they're saying is that there's obviously a linear relationship in the green field here between the dose of carcinogen and the tumor yield. However, humans would never be exposed to such high levels. Humans are exposed to a level shown by the red X. And they're assuming a linear relationship between the two. If there's a cancer risk here for the green levels, there's going to be a cancer risk here for the red levels. And so they assume that this linear relationship exists. But that's not always true. Sometimes there's a linear relationship between dose and tumor yield. But sometimes there's what we call a threshold. For a threshold, that means that you must reach a certain dose before that carcinogenic effect is shown. So in this case, if we took the same data points at the top here, took these same data points, right, that showed a linear relationship on the left, but if we actually did the studies where we used lower doses, we would see that it's not a linear effect, but instead there's this threshold where you must meet this minimum dose before you start to see any carcinogenic properties of the chemical. Some chemicals are like that, where it's not a linear relationship, but instead a threshold that must be met before the carcinogenic effects show up. And that's something that to be aware of when you see animal studies where they've given them large doses. It doesn't necessarily mean that there's a linear relationship at the lower doses. So let's look at some examples here, models for the relationship between the carcinogen dose and the cancer risk. 3 that have been identified so far as different ways that those two factors can be related. First, of course, is the linear model we talked about before, where you have this dose-response relationship. There's no threshold. It's a linear relationship between carcinogenic dose and the cancer risk. The threshold model, the other one we just talked about here, threshold means that there's no cancer risk at the lower doses, and you have to meet that threshold level. That level would differ for different chemicals, but you must meet that threshold level before you start to see a linear relationship between the carcinogenic dose and the cancer risk. The last model, which we haven't talked about yet, hormetic model, this is kind of an interesting one that is relatively new in cancer biology. And this is the idea that very low levels of the chemical actually can reduce the cancer risk. They're anti-carcinogenic. But once you increase the levels, they start to show carcinogenic properties. So it's a very interesting relationship. So low doses are actually decreasing your risk. Higher doses are increasing your risk. So there's a couple of chemicals that seem to follow that hormetic model, which is kind of an interesting one. The other question you always have to ask yourself when we're talking especially about animal studies is that animals are not all the same in their cancer risk, even when we're talking about the very same chemicals. So for example, there was a lot of data showing that saccharin caused bladder cancer in rats. No doubt you guys are aware of that you shouldn't eat artificial sugar products and artificial sweeteners. Saccharin was one of the big ones that they accused because of that risk factor that was associated with bladder cancer in rats. Interestingly enough, however, data shows that when you use the exact same chemical, saccharin, into hamsters, guinea pigs, and mice, you don't see a bladder cancer risk. And these are animals that are relatively closely related. Rats and mice especially are relatively closely related, and they don't show that same risk factor to saccharin. And that's actually when saccharin was taken off of the list of carcinogens in the United States, because we don't have data to show that it causes bladder cancer in humans. And we don't have reliable data to show that it causes it in a number of different animals. So it was taken off that list. Therefore, the U.S. government publishes lists of chemicals in two different ways. They publish what they call the known to be a human carcinogen list, and they publish what they call reasonably anticipated to be human carcinogens. So known to be a human carcinogen means they have evidence in humans. They have epidemiological evidence. Usually that started with animal model data, and then they also have data in humans to back up that risk. So known to be human carcinogens requires human data to show that it is a carcinogen. Reasonably anticipated to be a human carcinogen is extrapolated from animal studies. They have data in animals. They haven't been able to show it in humans yet. Of course, that's the limits of science there, but they anticipate it could be a carcinogen in humans. So that's the distinction, is whether or not they have human evidence to back it. All right, so before we get into the specific chemicals and how they cause cancer, it's important to talk briefly about what cancer is at a molecular level. And cancer really is mutation or change in the nucleotide sequence of DNA. Probably should reorder that slide so it says that. Change in the nucleotide sequence of DNA or mutation. Now, mutations are random. Very rarely do we see mutations occurring at a particular point unless there's a biological reason for that. So mutations randomly occur all over the genome, and they occur randomly in time. You can increase the frequency various different ways, including with a carcinogen, but they are normal random events, and they slowly accumulate with age. Mutations in germ cells, of course, would be passed on to future generations and to offspring. But mutations in somatic cells would only affect that individual organism. Now, those are the mutations that we generally think of as associated with cancer. So if you have mutations to the somatic cells, especially if you have mutations in some of these genes like P53 that we've seen before, that can result in cancer. And nearly all of the carcinogens are mutagens. There are some exceptions. Remember, alcohol doesn't cause mutations, but instead triggers inflammation. But nearly all of them are mutagens of some type. So that's kind of the bad news, is that mutations are constantly occurring all over the genome. The good news is that we do have repair mechanisms in order to repair these mutations when they occur. There's really two different kinds of repair mechanisms. There's what we call mismatch repair, mismatch repair will repair errors that come from DNA replication. They can be repaired by proofreading, which we've talked about polymerases do proofreading, or by what's called mismatch repair, and I'm going to talk more about that in a moment. Other types of DNA damage can be repaired by what's called excision repair, where you're going to cut out larger sections of DNA. So I'm going to go through both possibilities here. As a reminder, replication. The replication machinery, the whole group of enzymes there that are doing DNA replication, makes an error every 10 to the 7th nucleotides copied. That's an incredibly reliable system if you think about it. Only one nucleotide mismatch out of 10 to the 7th nucleotides copied. That's mind-blowing how good it is. Now these would be errors that escaped the proofreading mechanisms. Remember, here we're showing polymerases that add on the right nucleotide. Most of those polymerases also have a proofreading ability where they can cut off the last nucleotide if it's not correct. So this G was going to bind to T. It added it and then it took it off. So most polymerases do have proofreading ability as a 3 to 5 exonuclease. They are capable of checking themselves and removing a nucleotide that they identify as incorrect. And this type of repair mechanism works really well. So these would be ones that, one error per 10 to the 7th would have escaped proofreading because proofreading checks it during the actual synthesis of new DNA. Now, we do have a way of even triple checking that the right nucleotide has been added, and that's called mismatch repair. So this repair system I'm going to tell you about next, DNA mismatch repair, improves the accuracy to 1 error per 10 to the 9th nucleotides copied. So even more specific, getting the exact right sequence every single time. All right. Now, of course, the importance of fixing a nucleotide if it has been mismatched. Here they're showing you, here's the original sequence where you have an A bound to T. Of course, that's appropriate. Theoretically, these two halves would be split. Each half would be copied. You'd wind up with 2 copies of A to T. If you have a DNA replication error where instead of a new T here you have a C put in, that error would have escaped both proofreading and mismatch repair in order to be there. What would happen is in the next round of replication, on one side you'd wind up with the right base pairing, but on the other side, during replication, you'd wind up with a brand new permanent mutation, a brand new base pairing there in the DNA sequence. And of course, that mutation could be catastrophic to protein function of whatever the protein is from that gene. Now, these mutations do happen periodically, and if they are fixed before the next round of DNA replication, it's not a big deal. But if they aren't fixed before the next round of DNA replication, that's when you're going to start to have problems and that mutation becomes permanent. So it's pretty important to fix those mutations quickly right after replication because you don't want that DNA to be passed on if it's incorrect. You don't want to pass on that mutation to daughter cells. So here's how you can fix these mutations right after replication. Here's DNA mismatch repair, improving the accuracy of replication. So here they're showing us at the top of the picture here in red and orange the old DNA strand, the template strand in orange, the new strand that was just replicated in red, and they're showing a little mismatch here. They're showing as a bubble, that's the mismatch here. What will happen is these mismatch repair proteins are able to recognize the newly synthesized DNA strand, the red strand. They'll specifically recognize that. they'll actually bind to that newly synthesized strand, and they will cut out only the new strand, not the original template strand. That's going to allow the normal replication machinery, polymerases, ligases, to get in there and replace it with new DNA. So they repair only the newly synthesized strand by recognizing it right after replication. There are a couple of modification steps that happen after replication. So it's important for mismatch repair to get in there before those modifications happen. That's how they recognize new DNA versus old DNA. It's before the new DNA is modified. So they'll get in there and they will fix that. They'll pull out that mutated area and allow those replication enzymes to do their job and to replace it with new DNA. Let's see, did I get everything I wanted to say here? Mutations in genes that encode the mismatch repair proteins are really catastrophic, and they predispose people to certain kinds of cancers. There's a particular inherited mutation in those mismatch repair enzyme genes, where if you have that, it increases your risk of various different kinds of cancers, because you can't fix these mismatch mutations that occur during replication and aren't fixed by proofreading. So if you have that genetic disorder, it does raise your risk for a variety of different cancers, which is not surprising because those mutations can accumulate. All right. Now, what happens if the cell doesn't identify this mismatch quickly enough, right? Let's say the mutation, the change in the code got by proofreading, it got by mismatch repair that happens right after replication. Now what happens? There is another repair mechanism that can pull out these mutations. Often that repair mechanism is really for spontaneous DNA damage. So here, besides DNA mismatch, which are errors that occur during replication, DNA can also be damaged spontaneously. Now, how can that happen? Various ways for spontaneous DNA damage to occur. This would occur randomly. This would occur on both strands. Remember, mismatch repair only is targeted to the new strand, but this type of DNA damage would happen on either strand. Wouldn't matter, and it occurs randomly at any time. You can certainly raise your risk through various different exposures, but it does happen all the time. So the most common examples here of this spontaneous DNA damage, first is depurination. Depurination is the loss of purine, either an adenine or a guanine, one of these purine nitrogenous bases. Now, this really happens because of life in an aqueous world. Water is reactive, and hydrolysis reactions are relatively common in biology. And so occasionally, because you have the water molecules present, it is possible to actually cleave off a purine through this hydrolysis reaction. That's called depurination. If that happens, you wind up missing a base. You wind up with a deletion mutation here, where one of the bases is missing, and of course that's going to be a problem during replication, because you're going to have a frame shift mutation where everything will be shifted off. You won't get the correct protein made. Now, depurination reactions happen thousands of times per day per cell. Wrap your head around that. Thousands of times a day per cell. Random guanine and adenine nitrogenous bases can be cleaved from the DNA. This is not an uncommon event whatsoever. So of course we're going to need a way to fix this, because this happens from life in an aqueous world. Similarly, deamination reactions. Deamination reactions also are hydrolysis reactions. It comes from water breaking off, in this case, an amino group here from cytosine. When that happens, it actually turns cytosine most commonly into uracil. Excuse me. Now, of course, base pair rules, you know, C binds to G, A binds to T, and if you remember from RNA, U binds to A. So what's going to happen here is if you've switched it out from cytosine to uracil, you're going to wind up with incorrect sequence once it goes through replication because the U is going to bind to an A. You're going to wind up with the wrong sequence. So this is another important thing to fix for the cell. It does come about normally about hundreds of times per day per cell, again from life in an aqueous world. There are downfalls to life in an aqueous world, and these are two of them. The other type of spontaneous DNA damage that we see occurring are what are called pyrimidine dimers. Pyrimidine dimers occur when neighboring pyrimidines, either thymines or cytosines, bind to one another. This most commonly occurs after UV radiation. So ultraviolet light exposure will cause these neighboring thymines to actually bind to one another, which is going to create kind of a wrinkle in the DNA and can certainly cause mutations after replication. So another common mutation that can be fixed through these repair mechanisms I'm going to talk about, I believe it's on the next slide, yes. All right, so to fix some of the spontaneous DNA damage, you can use excision repair. Now, excision repair is exactly what it sounds like. You're going to excise the damaged DNA, and you're going to allow new DNA to be put into place, right? And the ligase will seal up the gaps. That's what excision repair is all about. Now, there are different possibilities here. Base excision repair will correct single damaged bases. So it will cut out just a specific nucleotide or a couple of nucleotides just right at one specific location, cut out that damage, and then allow the new damage to be filled in. Nucleotide excision repair will fix much larger lesions, so they'll fix pyrimidine dimers, or if you have a number of depurinations that have happened in one location, nucleotide excision repair will take care of those bulkier pieces. But in all cases, what's happening here is these repair nucleases recognize and remove that damaged region, and they are specialized for a particular type of damage. So there are excision repair enzymes that are responsible for pulling out pyrimidine dimers, and there are excision repair enzymes that are responsible for identifying deamination events. So they are specialized to their particular type of damage, and they will cut out that damage. And then they're going to allow the polymerases and ligases to get in there and fix that, replicate new DNA, I should say, to fill in that gap. And these are commonly found in all cellular organisms. Bacteria have them, we have them. They're found across all cellular life forms. Now, these excision repair enzymes are so important that when patients have mutations in their excision repair genes, it does lead to a much higher cancer risk. For example, xeroderma pigmentosa is a genetic disease where they have a mutation in the, it's called the NER system, and that is excision repair enzymes that specifically repair UV damage. Right, so xeroderma pigmentosa have these mutations specifically in excision repair enzymes that deal with UV damage, which is pyrimidine dimers. What happens is, if you don't have them, you cannot go out into the sunlight because UV damage happens, you get pyrimidine dimers, and if you don't have those repair mechanisms, you can't fix it. So you've seen probably, they've done shows on these kids that have xeroderma pigmentosa where they can't go outside ever, they have to play outside only at night, they have to wear hoods, and they have to wear gloves all the time, they can't have any contact with the sun because they don't have ability to fix these mutations should they show up. All right. So DNA damage, of course, can also be induced by environmental agents, which we're going to talk more about. Radiation, of course, exposure to UV light can induce more pyrimidine dimers. We'll see that X-rays and other types of what's called ionizing radiation can cause breaks in the DNA helix or can cause individual mutations. There are base analogs that mimic nitrogenous bases and then get incorporated accidentally into the DNA. Those generally are catastrophic to DNA replication. They'll often stop DNA replication if they get incorporated into the DNA. And there's base modifying agents that will alter the DNA structure. So an alkylating agent is one that adds a methyl group to the nitrogenous base. Some reactions with carcinogens will add these large bulky groups. I'm going to show you some examples in just a moment where they actually modify the nitrogenous base itself inside of the DNA structure and cause damage. The other possibility are what we call intercalating agents. Intercalating agents, they call that because they intercalate or sort of insert themselves into the DNA structure, and that causes often permanent damage. And so, for example, if you've worked with ethidium bromide in the past, it is an intercalating agent. It's commonly used to stain DNA once you've run a gel. But it is quite carcinogenic because it is an intercalating agent and it inserts itself into the DNA structure. That's how it causes its damage. All right. Let's talk about different groups of chemical carcinogens. Actually, all right, so different groups of chemical carcinogens here. There's really 6 main different chemical groups that can cause or have been identified to cause cancer in humans. On the top there I have polycyclic aromatic hydrocarbons, organic molecule of course. These have multiple fused benzene rings. You can see them in orange here. You can see those multiple benzene rings that help you to identify a polycyclic aromatic hydrocarbon. These are commonly found as natural components of tar and coal and soot and oils. Commonly have these polycyclic aromatic hydrocarbons. We talked about the smoke that comes when you're grilling meat and the juices fall down onto the grill and the oil that comes up. What it is are these polycyclic aromatic hydrocarbons, so associated with cancer, and they're usually found in these organic oils and tar and those sorts of substances. There have been some found in the incomplete combustion of tobacco as well, so it is part of what makes tobacco carcinogenic. Remember, there's a number of different carcinogens in tobacco, and so actually most of these are found there in some nature. Next one here is the aromatic amines. These are also organic molecules. In this case, they possess an amino group that's attached to the carbon backbone with a benzene ring or two. And so you can see them here, aromatic amines. You can see that benzene ring formation with an amino group attached to it. These have been found in a variety of different chemicals. The dyes that we talked about before, manufacturing of dyes, many of those are aromatic amines. Some of them are also found in tobacco smoke. Several aromatic amines are found in tobacco smoke, another carcinogen found there. Most of these aromatic amines are strongly carcinogenic. Some are not, but most of them are. You can't always assume something based on the structure, but most of them turn out to be carcinogenic. The next group here, the N-nitroso compounds. Again, organic molecules here that contain a nitroso group, which is nitrogen bound to, I think it's double bound to oxygen here is the nitroso group. Sorry, I'm just looking at my notes to make sure I'm saying this correctly, is how you identify them. Most of them are used in industrial or research chemicals, so they're mostly workplace carcinogens when you find them. There are a couple of the N-nitroso compounds that are found in cigarette smoke. Interesting, one that we already talked about does fit into this group, the nitrates and the nitrites that are found in meat curing and preservation. Remember, those are not carcinogenic themselves, but in the stomach, they're actually converted into one of these N-nitroso compounds. They're actually converted into a compound that can cause cancer. So they're ingested by themselves. They're not a problem. It's actually the transformation into this N-nitroso compound that causes them to become carcinogenic. An alkylating agent. Now, an alkylating agent, it's much harder to find sort of that reliable structure with an alkylating agent. What really makes this group a group of any type is the fact that they are molecules that kind of readily undergo these reactions where they attach to a carbon-containing group of another molecule. In other words, alkylating agencies like to attach themselves to other molecules. That's going to be how they cause cancer, is they're going to latch on to usually nitrogenous bases, and that's going to cause damage in the DNA, these alkylating agents of various different structures. The last of our organic molecules here in pink, the natural products. This is a very diverse group. There's not a particular structure that we can identify here. These are cancer-causing molecules that are produced by biological organisms. Mostly we're talking about microorganisms or plants that are going to make one of these natural products that we identify as a carcinogen. Classic example is one that we talked about, aflatoxin. Remember aflatoxin, which is caused by that mold Aspergillus. One of the most potent carcinogens that we know about, this aflatoxin, that can contaminate grains and nuts, and especially when they're stored under humid conditions. The last group here is the only one that has inorganic structure. The inorganic substances here. This is a handful of different inorganic molecules that have been identified as carcinogenic. Included in this group would be a group of metals such as cadmium, chromium, nickel, for example. They appear to be carcinogenic in both animal studies and human epidemiological studies. Now, I mentioned it with a couple of these groups, but I want to go back and kind of drill it home here, that some carcinogens, as they're ingested, are not actually carcinogenic. They're actually ingested in this sort of safer form, and then they need to be activated by a metabolic reaction before they exert their carcinogenic properties. Commonly those reactions happen in the liver or because of liver enzymes. That's not always true, but commonly that's true. And that's what they're showing you here in this picture. They're showing you in comes the foreign chemical in dark blue, a molecule coming from the liver called cytochrome P450. This enzyme really will activate that foreign chemical and cause some type of biological change in it, which eventually causes it to become carcinogenic. And that's true of several different molecules. So in their original ingested form, they may not be carcinogenic, but they can become carcinogenic because of reactions that would happen in our bodies. This is called carcinogen activation, this particular phenomenon. They would commonly call this foreign chemical, before it's carcinogenic, pre-carcinogen, right? Because it doesn't have carcinogenic properties on its own, but body enzymes will actually convert it into this more dangerous carcinogen through this process. Now, this process of carcinogen activation There are several different things that could happen to a molecule for it to become carcinogenic, but really the most common, really by far the most common type of reaction that happens is during carcinogen activation, this molecule becomes much more electrophilic or electron loving. So many carcinogens are electrophilic molecules, or if it was a pre-carcinogen, it got transformed into an electrophilic molecule. So again, electrophilic means electron loving, meaning that they want to react with substances that have a lot of electrons. So they are searching out molecules that have lots of electron molecules, or lots of electrons in them in order to attach to them, usually is what's happening. So for example, here's a reaction where this molecule is undergoing significant changes, and as it is, it's becoming more and more electrophilic. It wants to bind to electrons to fill some of these bonds. What happens is we have several different electron-rich molecules in our cells. DNA, RNA, and proteins are all electron-rich macromolecules. They have atoms in them that have lots and lots of electrons. It appears that DNA is a prime candidate for these electrophilic molecules to attach to, as opposed to RNA and protein, and that's what you're seeing here. This electrophilic molecule is attaching itself to a electron-rich nitrogenous base, in this case guanine. Unfortunately, when this binding happens, right, you can see here this binding of our electrophilic molecule to the guanine causes a distortion in the DNA, and this can result in cancer. It can block replication, it can cause replication errors, etc., by forming what's called a DNA adduct, where the molecule is binding to the base and then causing this distortion in the DNA structure. Many of the different carcinogens can form these DNA adducts where they attach themselves to the nitrogenous bases. And I have a quote here that despite their chemical diversity, many carcinogens share the property of becoming converted into electrophilic molecules, either in the body or before they enter the body. And that in turn causes them to be linked to DNA. And the ability to form these DNA adducts is one of the best predictors that a molecule can cause cancer, is the fact that it's extremely electrophilic. So it's a property we can see in many of these cancer-causing molecules. So a summary here of different kinds of DNA damage that can be caused by chemical carcinogens. You can get, we haven't talked a lot about this, I'm going to talk more about it with radiation, but single-strand DNA breaks, where you actually break the backbone of the DNA on one strand, or double-stranded DNA breaks, where you break both strands. These are more common with radiation. I'm going to talk more about that in just a moment. You can get links between adjacent bases. Remember, this is the pyrimidine dimer, where it can be between usually cytosines or thymines. You could get it cytosine to thymine, by the way, as well. It will work that way as well. Occasionally you see cross-linking between the strands, where you have it happening across here. This is damaging because it blocks replication. The strands don't separate the way they're supposed to during replication. Various different changes to bases. You can get missing bases because of a depurination reaction. Remember, there's the deamination reaction that can cause damages to bases and cause a change in the DNA sequence. Here's an adduct where you have this large molecule that was extremely electrophilic attaching itself to a base, and that's going to cause this distortion in the DNA sequence and eventual mutation as well. So lots of examples of how chemicals can cause these types of damage. All right, interestingly enough, when we look at data from various different chemicals, it turns out that true carcinogenesis requires 2 steps in order for it to happen. It requires what's called initiation and promotion. Interesting data that indicated that this was true, that you need 2 different steps. So we're going to look at an example here where they were studying tumors in rabbits, and they were using 2 different chemicals, DMBA, which is known to cause mutations, and croton oil, which they didn't actually know what it did at the time, except for that it seemed to raise the risk for cancer. So here's what they did. They would expose the rabbits to DMBA, right? And they would find only a small increase in cancer rates. But if they exposed the rabbits to DMBA and then later exposed them to croton oil, they would see a huge increase in cancer rates. But if they exposed them to no DMBA but only croton oil, they didn't see the increase in cancer rates. So obviously these 2 are working in a synergistic fashion. The DMBA and the croton oil are working together. And what's happening is DMBA will cause mutations. That's called initiation. We now identify that as initiation. That's the original mutation that comes about during carcinogenesis, that first step where mutations occur. Now, again, remember, if the mutation is fixed quickly, the mutation is fixed through proofreading, mismatch repair, excision repair, before the first round of cell division, no harm, no foul. Carcinogenesis only happens if that mutation becomes permanent. So initiation is the original mutation, in this case caused by DMBA. Turns out, however, that that's not enough to cause cancer in these rabbits. They need what's called promotion. Promotion will promote tumor development somehow. Turns out that croton oil stimulates cell proliferation. So it's really kind of a 1-2 punch. You get that mutation, and then you stimulate cell proliferation. And what you're doing is you're stimulating the proliferation of mutated cells. That's what was causing the increased cancer risk for these rabbits. So the DMBA on its own would cause mutations, and you'd see a very mild increase in cancer risks because of those mutations and natural cell proliferation. Croton oil on its own, right, you didn't see an increase in cancer risk because you didn't have the original mutation. So proliferation of normal cells wasn't going to cause cancer. But when you put the 2 together, if you have DMBA, which causes the mutation, and later you expose them to croton oil, which causes the proliferation of those mutated cells, that's when you wind up with large numbers of tumors in these rabbits. So it appears that carcinogenesis really requires initiation, the original mutation, and some ability to trigger cell proliferation, to trigger this growth and proliferation of these damaged cells. So we kind of put it together. This was the original idea, was initiation and promotion, but we also know of a phenomenon called progression, tumor progression, so I want to kind of put them all together here into one location. These 3 different steps that occur, sort of main stages of carcinogenesis. So the first one here, initiation. Of course, initiation is that first stage that is the original DNA mutation. Rather, it is an intercalation or pyrimidine dimer or depurination reaction or electrophilic molecule adducting to the DNA. Whatever it is, that original mutation is initiation. And they're showing you here, kind of in cream color, are the original healthy cells. One of the cells has undergone mutation. It's undergone that first step of initiation of carcinogenesis. The next step, of course, would be promotion. Promotion is the stage in which the initiated cell is stimulated to proliferate somehow. Now, promotion could occur through various different means. Get my arrow back here. Promotion could happen through various different means. It has to be something that's stimulating that mutated cell to proliferate faster than the other cells. You think about what that could be. Remember, alcohol can stimulate cell proliferation. Inflammation is really good at stimulating cell proliferation because it's the repair mechanism from an inflammatory event. You could have croton oil as stimulating promotion. You could have perhaps the mutation occurred in a gene that causes cell proliferation when it's damaged. So that you get kind of initiation and promotion in one double whammy. But somehow you're causing this that mutated cell to proliferate, and you have large numbers of it. Oftentimes, again, this is multiple mutations in order to get these proliferating damaged cells. And then progression. Progression here is really the theory that we've talked about before, tumor progression, where you get further mutations, further changes in gene expression that create these variant cells that exhibit sort of enhanced growth rates and other aggressive properties. Remember, this is natural selection. What happens is those that are mutated to divide the fastest eventually begin to take over and predominate, and that's tumor progression. Over time, the tumors become more aggressive, more likely to metastasize, more likely to resist cell cycle signals and those sorts of things. So that's progression. Now we identify those 3 stages that happen in carcinogenesis: the original mutation, something that's going to stimulate proliferation of that mutated cell, and then eventually tumor progression, where the tumor is sort of evolving to better survive in its environment, which is unfortunately is our cells, and to eventually become more aggressive through that evolutionary process. All right, the next topic I want to talk about is radiation in cancer, which goes really kind of hand in hand with what we've been talking about so far with chemicals. But hold on for a second, I'm going to grab my oatmeal. All right. Radiation and cancer. Radiation is defined as any type of energy traveling through space. That could be heat, that could be sound, that could be radio waves, microwaves, visible light, ultraviolet light, X-rays, nuclear radiation, etc. So any energy traveling through space would be defined as radiation. That said, the greatest cancer risks are associated with exposure to high energy forms of radiation because they have to impart enough energy to alter a chemical bond. So I'm not going to talk about radio waves and microwaves and heat and visible light because they're low energy and we don't have any evidence to show that they're associated with an increased cancer risk. So we're going to stick to the ones that are higher energy and do show increased cancer risk. We'll start with ultraviolet radiation, which of course comes from the sun. Ultraviolet radiation is responsible for the majority of all cancers. In fact, half of all diagnosed cancer cases are skin cancer. So it has kind of the dubious distinction of being the most common form of cancer are these skin cancers. UV radiation is responsible for almost all of those skin cancers. Interesting, we can associate, sorry, I'm eating my oatmeal while I talk to you. We can associate skin cancers with UV radiation exposure. The more exposure you have, the higher your risk for developing skin cancers. This is a map of the United States showing sunlight intensity. And red here in this southwest areas where you have the highest sunlight intensity, and green, the very, very tip of Washington State, there is the lowest. And you see in yellow a number of low areas in the northeast as well. And it's probably no surprise to you to find that in areas of the northeast here, you see relatively low cancer rates, two different common kinds of cancer we're going to talk more about in a moment. Relatively low rates in these areas where they don't have a lot of sunlight exposure versus much higher rates in New Mexico and parts of the United States where you have a higher exposure to this UV light. A couple of other interesting bits and pieces. Definitely is associated with people who have fairer skin. Turns out that melanin, which is the pigment in our skin, does absorb UV light. So the more pigment you have, and the more melanin you have in your skin, the more UV light is naturally absorbed by the melanin. It does decrease your skin cancer risk. Not to zero, but it does decrease your skin cancer risk. Without a doubt, the part of the world that has the highest risk for skin cancer is Australia. Australia has an extremely high risk for skin cancer. It's not particularly surprising because, and really I should be more specific, because it's really the white population in Australia that has such a high skin cancer risk. Fair people from England immigrated there. High levels of sunlight exposure. It's a bad combination, fair skin and high levels of sunlight exposure. So they definitely have the highest risk anywhere that we've seen. Another interesting factoid about sunlight and skin cancer is that skin cancer normally affects areas that are generally uncovered by clothing. So you see skin cancer risk highest on the face, the chest, the arms, not on the belly or those sorts of areas. So it's parts of the skin that are commonly exposed to the light where we see the greatest risk for skin cancer. All right, so more specific on skin cancer here. The incidence and mortality rates for different types of skin cancer. The pie chart on the left there is cancer cases. Pie chart on the right is cancer deaths for different kinds of skin cancers. First here is basal cell carcinoma, and you can see that that is by far the most common type of skin cancer. Over 3/4 of all skin cancer cases really come down to them being basal cell carcinoma. Now, basal cell carcinoma is extremely common, but it does not metastasize well. In fact, it metastasizes very, very poorly. Only 1 out of every 1,000 cases of basal cell carcinoma have metastasized. So extremely low rates for metastasis, and that's why it is the most common cause of skin cancer. But it's almost unheard of to see it causing cancer deaths because it doesn't metastasize. It's quite easy to remove quickly from the surface of the skin, and it almost never metastasizes. Next one here, squamous cell carcinoma. Squamous cell carcinoma, second most common form of skin cancer. It still is not great at metastasizing, better than basal cell carcinoma, but still not great. Squamous cell carcinoma metastasizes about 1 out of every 20 cases, so it is responsible for a proportion of skin cancer deaths here, but again, it's not a common metastasizing cancer. Really kind of the baddie in this group is melanoma. Melanoma is a cancer that arises in the melanocytes, and unfortunately, well, actually let me say fortunately first. Fortunately, melanoma is the minority of skin cancer cases, of diagnosed skin cancer cases is melanoma. Only about 5% of skin cancers, wait, I'm sorry, I think I'm looking at the wrong statistic. Oh yeah, no, that's right. Only 5% of skin cancers are melanoma, so it is a relatively rare form of cancer. And unfortunately, melanoma is known for its ability to metastasize. In fact, melanoma can metastasize so quickly that it often has metastasized even before the patient gets diagnosed. It's really good at metastasizing, and that's why melanoma is the major cause of skin cancer deaths. Even though it's a relatively uncommon form of skin cancer, it's responsible for the most skin cancer deaths because of its ability to metastasize so easily. All right. All right, so what's in the sunlight that's causing this risk for skin cancer, of course, is UV radiation. If we look at radiation here, it's all sorted out according to wavelength. Generally speaking, the shorter the wavelength, the higher the energy. Higher the energy, more likely it's going to cause damage to chemical bonds, including DNA damage, and eventually can result in cancer or other types of damage. So here's visible light. The visible light spectrum here, shorter than visible light is UV light, right? Shorter wavelength, higher energy, more likely to cause damage. X-rays are even shorter, and gamma rays are even shorter, so we'll talk about those as well. Really, no risk has been associated scientifically with any of these longer wavelengths. So we don't see anything with radio waves, cell phones, microwaves, infrared. We don't see an increased risk for cancer. It's not surprising since they are lower energy types of radiation. It's not surprising that they wouldn't show an association with cancer or tissue damage. All right. 3 different types of UV radiation that comes from the sun. You can see it split up here into UVA, UVB, and UVC. A has a longer wavelength than B. B has a longer wavelength than C. So the shortest wavelength is UVC light. Now UVA. UVA light is not filtered by the atmosphere. It does have the longest wavelength. The wavelength for UVA light is somewhere between 315 to 400 nanometers is the wavelength for UVA light. We don't have a lot of data to show that it's associated well with skin cancers. However, it does seem to be associated with aging. Some data indicates maybe it's associated with skin cancers, but really the strongest association so far has been aging of the skin, and long-term damage to the skin that comes from sunlight is probably coming from UVA rays. UVB rays. UVB rays are somewhere between 280 nanometers to 315 nanometers. They are mostly absorbed by the ozone that's in the atmosphere. Some of them, of course, are not, but mostly it's absorbed by the ozone that's found in the atmosphere. When they penetrate through the atmosphere, they cause tanning, aging, burning, and they're associated with cancer. Make sure you shut the door, hon. And they are associated with cancer. UVC rays. UVC rays are completely absorbed by the atmosphere. They do come from the sun, but they do not penetrate through our atmosphere and enter into onto Earth. However, some can come from artificial light sources. So, for example, sterilizers that you find in a micro lab or other environments, things that are used to sterilize equipment, do emit UVC rays. So you can wind up with damage from UVC rays even though they don't come through our atmosphere from other sources. UVC seems to cause extremely severe burns. That's the major component of damage that comes from those UVC rays. So kind of the biggie here that we're going to talk about are UVB, because those are the ones that cause most of the damage associated with skin cancers. So let's look at how UVB rays can cause damage to the DNA. Really, we kind of already touched on this topic, but we'll come back just for clarity's sake and put it in the right spot here. UVB radiation creates pyrimidine dimers in the DNA. Remember, pyrimidine dimers are those bonds that happen between neighboring cytosines or thymines, or cytosine to thymine, adjacent I said that kind of unclearly. Let me say that more clearly. So two adjacent cytosines or thymines can bind together, forming what we call a pyrimidine dimer, which causes distortion in the DNA and can cause a problem during replication. Following along in this diagram, you see in the base pair sequence, there's two cytosines here, of course, base pair, two guanines. UV radiation causes the neighboring cytosines to bind to one another, forming this pyrimidine dimer. During DNA replication, the two strands are separated. The G's here go on to make a healthy new DNA molecule and cell, but the C's, this distortion in the DNA actually will stimulate incorrect base pairing. DNA polymerases cannot read those pyrimidine dimers very well, and that will trigger incorrect base pairing here. So you can see two adenines were added in. When that goes through replication, you're going to get the incorrect sequence on both sides. So it's problematic because those pyrimidine dimers, if they are not fixed, will cause incorrect base pairing during replication. Now again, if repair happens early enough, excision repair happens early enough, that would fix this problem, but if it doesn't happen before replication, the mutation becomes permanent, and that's the cause of damage. Now, I mentioned, well, more than mentioned, I talked about mismatch repair previously. I want to remind you that mismatch repair happens all the time, but if you have enough of these pyrimidine dimers, what happens is you sort of overwhelm the ability of these excision repair enzymes to do their job, and that's what eventually can cause skin cancers and these permanent pyrimidine dimers. So the base substitution that happens at adjacent pyrimidines is a signature of UV damage because you had the two pyrimidines, right? If you have two pyrimidines next to each other and that mutation has happened there, that's the signature of UV damage, indicating that it came from a pyrimidine dimer caused by UVB exposure. Some more information here. Incidence of two types of P53 mutations in skin cancer and internal cancers. Remember, P53 is the most common mutation to find in cancerous cells, or it is the gene that's most commonly mutated in human cancers. If we look at two different kinds of mutations that could occur in P53, so on the left side here we're looking at mutations that were cytosine and got mutated to thymine. This is a signature of UV damage, right? Remember, two pyrimidines next to one another that have been mutated is a signature of UV damage here. Now we're looking at another type of mutation that can occur in P53 for comparison's sake, and here it's one cytosine at one location that has been mutated to thymine, not two of them. So this is not caused by UV damage, right? So here on the left we're looking at UV damage, here on the right we're looking at another kind of mutation not associated with UV damage. And what they find is that if you look at skin cancers, this UVB signature damage mutation is incredibly common in skin cancers. In fact, it is the major cause of skin cancers. But if you look at internal cancers, almost none of them have this UVB signature. So UVB light is associated with skin cancer, but not internal cancers. Likewise, if we look at this other random mutation, you almost never see it in skin cancer, but you do see it periodically in internal cancers. So what I'm trying to prove to you with this picture is that UVB light causes the vast majority of skin cancers through the formation of these pyrimidine dimers. It's almost exclusively UVB light that is doing that. It's causing the pyrimidine dimer mutation and eventually arising in skin cancers, but not internal cancers. So if we kind of map through the process of how this would work, so sunlight-induced P53 mutations and the development of skin cancer. Here in cream we have the healthy skin cells here, the outer layer of skin. UV radiation from sunlight would cause DNA mutations. Now, does the sunlight like directly go to P53 and cause a mutation right there in that particular gene? No, it's random, right? The pyrimidine dimers would form randomly in the genome. They could form at multiple different locations. So here in purple, they're showing you all sorts of different mutations that could happen because it happens randomly in the genome. Now, they're showing you in purple, it's happened at random locations in the genome. This kind of, I don't know what color that is, scarlet? At any rate, this particular cell, it happens to have happened in P53. The pyrimidine dimer happens to have formed in P53 out of random chance. Now remember, a damaged cell, a cell that has a mutation, is going to trigger apoptosis. It's a normal process in the cells in order to get rid of DNA damage. So you would expect all of these purple cells to die through apoptosis. They've been damaged. That's the whole point of that backup system. But of course, our scarlet cell that has been, it's not really scarlet, I don't know what color to call that. Anyway, this cell that has the P53 mutation can't go through apoptosis because it doesn't have an active form of P53. That's going to mean that it can be proliferated and you're going to start to see additional cells with P53 mutation aren't going to go through apoptosis, and eventually that leads to the development of skin cancer. So again, mutation is not directed at one particular gene. It happens randomly. If it happens to occur on something like P53, the problem is it's not going to be killed off. That cell won't be killed off the way that other cells would be after DNA mutation. So the good news, however, about skin cancer is that it's relatively easy to protect yourself against skin cancer. Probably easier than other forms of cancer. Because we know that skin cancer arises from exposure to UVB light. So the first one, seems sort of obvious, is avoid exposure to UVB light. Oh my god, he's cold. So avoid exposure to UVB light. Don't go outside in the hottest part of the day. You want to wear hats and long sleeves, those sorts of things, to avoid exposure to the UV light. In Australia, they had a whole campaign they called Slip, Slop, Slap, where they told you to slip on a shirt, slop on some sunscreen, and slap on a hat. And that was their campaign to try and reduce skin cancers in Australia. Now, sunburn is actually a normal, natural protective mechanism for dealing with UV damage. It's actually, sunburn happens to trigger the destruction of the outer layer that's been damaged. That peeling off of skin and all of that stuff is there to try and get rid of any possibly damaged cells. Unfortunately, the UV light can go a little bit deeper and you may not get rid of all of those mutations. Sunscreens work really well. Some sunscreens reflect UV light, so the original ones like zinc oxide, things like that, they actually reflect the light. Some of the newer ones actually absorb it. In either case, they're either reflecting the radiation or absorbing the radiation so it doesn't get to your skin. Work very well, as long as they're used properly. Melanin in the skin absorbs UV radiation and prevents damage, which is why people with more melanin in their skin have a lower skin cancer risk. It does not go to zero, but it is a lower skin cancer risk than those that don't have much melanin. Some people think that because of that, tanning would be a good idea, because tanning does seem to overexpress melanin in the skin. So the idea being that if you kind of get that base tan, you're going to have more melanin and it decreases your skin cancer risk. Interestingly enough, however, they find the exact opposite to be true. Tanning uses both UVA and UVB light. And UVA and UVB light together seems to raise the risk for skin cancers. And they don't have a very good explanation yet of what's going on. Certainly, UVB light exposure raises skin cancer risk. UVA light exposure causes aging to the skin. gives you that kind of leathery look. And together, tanning raises the risk of skin cancer. It does not lower the risk. All right, let's talk about another form of radiation here. We're going to talk about ionizing radiation. Now, ionizing radiation, ionizing means that it removes electrons from a biological molecule. So ionizing radiation removes electrons from a biological molecule. When you do that, you generate a highly reactive ion that can damage the DNA, right, when you take off those electrons. So we're going to look at several different examples of ionizing radiation, starting with X-rays. But in all cases, what that means is it's forming these highly reactive DNA damaging molecules because it's removing electrons from these biological molecules. So, all right, X-rays. X-rays are kind of the classic in this group. X-rays do have shorter wavelength than UV light, which means that they are at higher energy and therefore can cause damage to chemical bonds. Now, the history here for X-rays, when X-rays were first developed, it was sort of a novelty to use these X-rays for everything, and people would go in and have like bone portraits done where they would have pictures of their bones and display them in their homes. So these X-rays would be taken. And it was sort of at first seen as a beautiful art form. Of course, scientists were using it and physicians were using them for diagnostic purposes, but it was sort of a novelty item at first. Thomas Edison's lab technician, he worked a lot with X-ray devices and X-ray equipment, and he routinely would test his X-ray equipment on his hands. So when he was testing equipment, he'd X-ray his hands. Just that's what he did all the time. And what happened is he eventually got these severe burns and then tumors on his hands from the X-ray, the ionizing radiation exposure. Eventually he became a double amputee. They had to cut off hands and then actually up to his arms. And eventually he died of, I don't even have the type of cancer. It was metastasized, but the cancer metastasized, and he was the first to be a death known to be caused by X-rays. And indeed, we know that radiologists have a higher cancer risk than the general population and people who work around X-rays. So definitely, here they're showing you dosage of X-rays and how much shorter your lifespan is. So scary for X-rays. Another type of ionizing radiation here, radioactivity. Radioactivity is radiation that is emitted by a chemical element that is unstable. So the chemical itself is unstable, and that's emitting this radioactivity or nuclear radiation, same thing. 3 different kinds of nuclear radiation. Alpha particles. Alpha particles are positively charged. We look at an alpha particle, it's 2 neutrons and 2 protons. So it's really a helium ion is what we're looking at here, and that is an alpha particle being released from these radioactive elements. Beta particles are electrons. Electrons that are released from ions, from these radioactive ions, are called beta particles. And then gamma rays. Gamma rays are a type of electromagnetic radiation that can be emitted from some chemical elements. Gamma rays, if you look here, have an extremely short wavelength. Gamma rays are really, really short in their wavelength. So hopefully it's no surprise to you that that means that they can cause significant DNA damage, because the short wavelength means high energy and they can cause significant DNA damage. All right, and of course Marie Curie was a discoverer of some of these gamma ray emitting radioactive elements, including radium and polonium, and indeed she died of leukemia because of her exposure to those gamma rays. Now, measuring radiation dose. And I know that this slide looks intimidating, but I'm going to walk you through, so don't be nervous. How we can measure your dose of radiation. Now, cancer risk is associated with nuclear radiation anyway, is directly related to the dose received. Not surprising, right? More radiation dose, higher radiation dose that you receive, the higher your risk for cancer. Not particularly a surprising factoid here. How we measure that amount of radiation. I'm going to kind of walk you through here. An electron volt. This is a term that a physicist would use. Maybe a chemist, but really a physicist is a physics term. An electron volt expresses the total amount of energy present. So that chemical that's emitting radiation, how much is it emitting? That is measured in electron volts. That's not particularly helpful to us, however, because we don't really want to know in biology, and especially cancer biology, what we're more interested in is how much of that energy is absorbed by a biological molecule or a biological organism. That's measured in grays. Grays describe how much energy is actually absorbed. This is a more useful number for a biologist who's looking at the effect on biological organisms, is to look at grays, how much is actually absorbed by that molecule. We can add in a correction factor. This is called relative biological effectiveness, RBE. This is a correction factor that indicates the damage to biological tissues. For example, you know that the shorter the wavelength, the higher the energy, the more damaging it is. Well, this RBE takes that into account. It indicates how likely that is to cause damage. So you take the gray, which is how much energy is absorbed, and you use this correction factor, the RBE, which is going to say how likely is it that that absorbed energy is going to cause damage in a biological system. You put it together to get what we call the sievert. And the sievert is what we usually talk about in biology because that's the really useful information. The sievert is the biologically equivalent dose. So I kind of went by this one quickly. I want to go back here. The relative biological effectiveness is a correction factor that indicates the damage to biological tissues. X-rays have an RBE of only 1, while an alpha particle has an RBE of 20. Which indicates that alpha particles are 20 times more effective at causing tissue damage, and that turns out to be because they have a shorter wavelength. That's why that RBE becomes useful, as it talks about how likely it is to cause the damage. And you put it all together, the gray and the RBE to get the sievert, the biologically equivalent dose. That represents how much biological damage is given during the exposure. And that's the kind of information that we usually use in a biology class, especially, because we're interested in damage to the organism and to the biological molecules. So sieverts really give a better indication of how much biological damage is given during an exposure to that radiation. All right. Alpha particles. Where do we find alpha particles? Alpha particles are very hazardous. They are very dangerous. However, alpha particles are easily blocked. They don't penetrate the skin. They don't penetrate through very much. They actually are very easy to block. The problem comes if they are inhaled or ingested. Alpha particles that are inhaled or ingested are a totally different case. That's when you start to see damage that happens at the biological level. The biggest risk here would be from radium. Radium is found in the ground and it is released as a gas. We call it radon. It's the gas that's released from the earth. You can see here places in the United States where they have high levels of radon gas being emitted from the earth. Unfortunately, what happens is it can cause mutations in lung cells, and it can then lead to lung cancer from long-term exposure to this radon gas. This really isn't an issue for most individuals. Where it can become an issue is for people who work or live in buildings that have very bad ventilation. Radon gas can accumulate in that building. You actually get higher and higher levels as the gas accumulates in that inside. So it's actually safer outside because it doesn't accumulate in these tight spaces with this radon gas that can cause damage. If we look at another type of ionizing radiation here, nuclear explosions and ionizing radiation. Nuclear explosions release a number of different kinds of, oh, I missed a great story. Let me go back. I'm sorry, I want to tell you this story. It's a great story. Well, I guess great in kind of a twisted way, but in the 1920s, a factory in New Jersey employed a bunch of women, and what they were doing is they were painting the dials on watch faces. They were making glow-in-the-dark watches, and they would use radium to make these glow-in-the-dark watch dials. And so what they would do is they would take this fine paintbrush and they would dip it in the radium, and then they would paint the surface of these watch dials in order to make them glow in the dark. And of course, if you've ever painted before, you're familiar with the phenomenon of your brush getting kind of yucky at the end. And so what you do is you just kind of lick it, dip it in your mouth, lick it, to pull those brush fibers clean. So what was happening is they were constantly dipping their brushes in the radium, painting the watch dials, and then putting them in their mouths. And the number of women from that factory that got bone cancer was astronomical. And that was actually one of the first times that they could show a cause and effect relationship using epidemiological studies between radium and cancer. So I guess it's not a good story. It's kind of a terrible story. But anyway. All right. Looking at nuclear explosions and ionizing radiation. This is ionizing radiation in massive doses. So here we're looking at cancer cases on the left and cancer deaths caused by excessive exposure to this nuclear explosion with ionizing radiation, shown in sieverts here, and they're showing huge numbers. So leukemia was at really high numbers from nuclear explosions. We then started to see it in other types of cancer as well. Leukemia is a relatively quickly developing cancer, so this was sort of the first one that was seen, but really many different kinds of cancers showed up. It wasn't just leukemia. It's just that one looks really extreme because it's such a quick form of cancer to develop. If we look at cancer deaths, you can see death rates are much higher for people exposed to that type of massive dose of ionizing radiation. We can even use a dose response curve looking at radiation exposure and cancer rates, specifically after the atomic bomb was dropped in Japan. So what we're looking at here are two different forms of cancers. Well, really leukemia, which is a cancer of the blood, and all others, the solid cancer tumors. We're looking at radiation dose in sieverts. This had to do with how close you were to where the bomb was dropped. Further away that you were, the lower your sievert dose. Closer you were, the higher your sievert dose. And what they're finding, of course, is a really clear correlation between increased dosage, in other words, closer to the explosion, and your risk for various different kinds of cancer. And that's no surprise because this ionizing radiation causes DNA mutations, which then result in higher cancer rates. Question I always get here, and it's the same question I have, is you see a peak, and then you see a drop, What is going on with this peak and drop? So, it's been postulated by the scientists who did this experiment that what was happening is that these higher doses may actually kill off the cells. So this particular dosage was high enough to kill the cells that were mutated, and so you didn't see that higher risk of cancer. I cannot explain to you, however, why it goes back up here. That's unclear. So, but there's some question as to if you're in this really high, extremely high radiation dose, does that somehow actually drop your cancer risk because you're killing off excuse me, you're killing off those cells, and you don't wind up with cancer coming from those cells. They have very similar data also after the 1986 nuclear power plant breakdown in Chernobyl. We have very similar data showing that how close you were to the Chernobyl facility raised your radiation dose and raised your risk for cancer. As an aside, thyroid cancer in children that were in that region is about 100 times higher than it is for children in other parts of Russia. So extremely high rates for that particular type of cancer. So what kinds of mutations are caused by ionizing radiation? A couple of different things have been identified. On the left-hand side here, I want to remind you that ionizing radiation means that it strips away electrons, which makes a highly unstable ion that can damage chemical bonds. And that can result in several different kinds of damage. It can result in base damage, where you have alteration of a base. You have this highly reactive species that will alter a base to cause damage, or complete hydrolysis. It will cut off this entire base. Excuse me, and you wind up with a deletion mutation. That's possible from ionizing radiation, causing these free radicals that can be extremely reactive and cause these bases to be eliminated or somehow altered. The other possibility is causing a break in the actual backbone of the DNA, either a single-stranded break or a double-stranded break. Can result from these ionizing radiation sources. Now, single-stranded breaks are relatively easy to repair accurately because you still have one strand bound, so you can repair that relatively easily. Double-stranded breaks are much harder because you have to line up the correct ends, and so there's a much higher error rate in repairing those double-stranded breaks. And those are a hallmark of ionizing radiation, are these breaks in the actual backbone structure of DNA, single-strand or double-stranded breaks. So if we look at overall sources of annual exposure to ionizing radiation in the United States, so how likely are you to encounter these different ionizing radiation sources? We measure these in BERTs. Like Bert and Ernie. So BERT stands for the background equivalent radiation time. Right? In other words, a BERT is your radiation exposure. It has to do with, see, I have it written out here, converts the dose of ionizing radiation to the amount of time it would take a person to get that same dose from a normal natural background source. So we're just comparing, a normal person living on the earth with a normal risk factor. If we compared this activity to that person, how long would it take to get that same exposure? I think this is most easily understood if we go through the examples here. So for example, if you take a transatlantic flight, right? Take a transatlantic flight, you're up in the air, you're closer to these ionizing radiation sources. You do have trapped air in that environment, which has a higher radon value, and your BERT is 1 week. So you get basically 1 week's worth of normal ionizing radiation from taking one flight. And indeed, pilots and flight attendants do have a higher cancer risk than the average population because of this. If you live near a nuclear power plant for a year, your BERT goes up to under 1 day, so not a high risk factor really. So a whole year you get less than a day BERT exposure from that. Chest x-ray BERT. From having a chest x-ray, you get 10 days of this kind of regular exposure level. If polonium in cigarettes will give you a BERT value of about 10 years. So once again, the moral to my story is don't smoke. So it kind of gives you an idea of the normal radiation exposure that you have and what sorts of things increase that radiation exposure risk. If we look at the normal annual exposure for the U.S. population, we see that over half of it comes from radon. Remember radon gas coming from the Earth. About 8% comes from cosmic rays, 8% comes from terrestrial environments, 11% comes actually from our own bodies radiating these different kinds of radiation, making natural sources 81% of our ionizing radiation that we encounter. If we look at artificial sources, medical x-rays are the number one. Other medical visits and various consumer products can cause these radiations, ionizing radiation. They account for about 19% of our risk on an annual dose. So total annual dose of about 3.6 millisieverts is what we see for a normal human in the United States and their exposure to ionizing radiation. All right, now the other thing that I just want to mention here is if you have the same amount of ionizing radiation exposure spread out over years, so if we say 10 millisieverts, if you have 10 millisieverts of radiation exposure spread out over multiple years, and then person B has 10 millisieverts exposed over the period of just a day or two, the risk is much higher for someone who has quick exposure. So exposure over years is less damaging than a high exposure over a short time period. Not particularly surprising because remember our repair mechanisms will get overwhelmed if that exposure is happening really quickly and they won't be able to fix the damage. All right, that brings us to the end of the chemical and radiation lecture. So I will see you next time.